and welcome to the webinar on comparative genomics as part of our webinar series. This webinar was originally broadcast by Astrid Gall yesterday evening, but we lost the video unfortunately, so I'm re-recording. So I'm covering comparing genes and genomes with Ensembl compara. Previous to this was a webinar from Erin Haskell on variation data in Ensembl and the Ensembl VEP. So what I'm going to cover today is what comparative genomics is used for, what species we have data for. I'm going to talk about gene trees and how these are used for homology predictions. And I'm going to talk about whole genome alignments, both pairwise and multiple. And I'll also cover shared synteny. So comparative genomics allows us to understand vertebrate evolution. This helps us to understand the differences and the similarities between species at the genome level. It also allows us to infer gene function based on homology, and it helps us to identify highly conserved regions, which are important in identifying what regions might be functional. We carry out comparative analysis on a per taxa basis. So if you're using the main Ensembl vertebrate site, we carry out a comparative genomics analysis on all of the genes in the set found in there. If you look at Ensembl metazoa, you get a similar thing but for all the metazoa, and as you go through the different divisions of Ensembl, there'll be a gene tree analysis and homology analysis for each one. We also have what we call pan-taxonomic compara, and this takes a subset of species from each of the divisions of Ensembl and does comparative analysis with this subset. And if you're looking at some of the species which are involved in pan-taxonomic compara, this is just the gene tab for mosquito, and you can see that there are actually two categories here. There's metazoa compara, which will give you gene trees containing other insects and worms and other kinds of non-vertebrate metazoa. And there's also pan-taxonomic compara, which will give you the gene trees and orthologs across this taxonomic space. Gene trees are computed based on protein alignments. So we choose a representative protein for each Ensembl gene. These are then clustered using BLAST to create multiple alignments, which are then reconciled against the species tree. And this allows us to infer the orthologs and the paralogs. Orthology and paralogy is defined by whether the thing that separates two genes is speciation or duplication. So if we look at this particular gene tree, paralogs emerge through a duplication event. So here we have this red duplication event, and so we can say that genes on either side of this duplication event are paralogs of each other. Orthologs emerge through a speciation event. So anything on either side of a blue blobby can be considered to be an ortholog. And these are split into one-to-one orthologs, so we can say that C1 and C2 C1 and H1 are one-to-one orthologs. Whereas the M gene, because there's only one copy of it, is a one-to-many ortholog. With human, there's H1 and H2. So we're going to have a look at a gene. We're going to look at BRCA2, and we're going to in human, and we're going to identify its homologs. So we're on the Ensembl homepage, which we've explored in some of the other webinars. So you should be nice and familiar with this by now. I'm going to just type BRCA2 into the search box and hit go, and go to my gene page. So the section we're going to explore in the menu is over here on the left, and the first thing I'm going to explore is the gene tree. So this is a graphical display of the gene tree. We can see our human BRCA2 highlighted in red here, and we've got speciation nodes shown in blue and duplication nodes shown in red. We can see that some of the nodes are expanded. Anything that leads up to this human gene are expanded, but some of them are collapsed, such as this node here with the Old World monkeys. If I click on this node, I can say expand this subtree, And now I have an extra bit of tree displaying all those individual Old World monkeys and the BRCA2 gene in them. If I look over on the right hand side of this image, we have these kind of green graphics. These are alignments. So we can see here's our BRCA2, and the green colour indicates where this is aligned to other BRCA2s in other species, and the white here indicates where it's not aligned. Now some of the, you can see some of these are different shades of green. Wherever we've just got a single gene, where it's an expanded subtree, it's this pale green. Whereas where we've got a collapsed node, such as this chimpanzees, we have this darker green. It's better to see these bony vertebrates here. We've got a mixture of the lighter green and the darker green. If I scroll down to this legend here, I can see that in these, within these nodes, if over 2/3 of the proteins in this node are aligned, it's dark green. The light green means that between 1 and 2/3 are aligned, and the white means that 0 to 1/3 are aligned within this. We also have some dubious nodes because we are reconciling against the species tree. There are some things that don't quite look right compared with what we understand of evolution. We can also download this tree. So we have a link here, this little download button. If I click on this, it will allow me to download the gene tree in numerous different formats. So I can download something like FASTA and just get all the sequences in the gene tree, but I can also download different kinds of tree formats, such as Genomic and things like that, that I can then put into a different kind of software. Another thing I can do if I go to a node, and this can be an open or expanded or a collapsed node, if I click on this, I have the option of view subtree in Wasabi, which is a really nice software for viewing protein alignment. So this is gives me the tree over here on the left, and I can see the sequences aligned with nice highlights and colours, so it's really obvious here that we've got a difference in this, I think it's orangutan. So, and also where we've got this sort of block of species here which have got something different, and we can see if we go back to our tree that they're kind of separate from the rest of it. So it's a really nice way to view alignment, and it can be edited how you like as well. I'm going to close Wasabi and go back into my browser, and I'm going to close this taxon here, and I'm now going to explore orthologs. So over here in the menu on the left, there's orthologs and paralogs. Because BRCA2, it doesn't have paralogs, that's greyed out, but if there were paralogs available, this would be coloured in blue. I'm going to click orthologs, and you can see we've got a table summarising how many of each kind of ortholog we have in different taxonomic groups. So there's of the primates, there are 22 with 1-to-1 orthologs, and if you're not sure what the terminology is, please just put your mouse over the headers and it will tell you. None of them have 1-to-many, and none of them have many-to-many. But there is one which doesn't have, one primate species that doesn't have an ortholog. So I can narrow it down using these boxes here. If I click on this rodents, I will only see the 24 rodents we have. Another thing I can do is I can just search for my species of interest over here using this box on the right. And you'll see we have a table that lists the species, the type of orthology. There's a link here to go to view the ortholog in Ensembl. You can see the identity. These GOC and whole genome alignment coverage scores are really good for indicating how likely it is that these are real orthologs, as opposed to possibly it could be a paralog, and not really sure. But in this case, we've got scores of 100 for GOC, for most things, and for WGA, very close to that. So it indicates this is a very strong ortholog between many of these species. If I look at mouse here, I have the option here to view sequence alignments, which is quite useful. So if I go into view protein alignment, there's also view cDNA. I get a cluster W alignment, which has got the human protein, gives the ENSP of it, and the mouse protein underneath. And if you're not familiar with cluster format, what these different symbols mean, it's all here in the help for this page. It tells you what they all mean. There's the percentage identity. You can see that that there's different identity depending on what your base is, whether you're starting with human or starting with mouse, because obviously they're slightly different length, which we can see here as well. So I'm going to go back to my presentation now, and I'm going to carry on talking about whole genome alignment. So whole genome alignments are really useful. They help you to identify highly conserved regions. These may be sequences that evolve slowly. It is likely that they are functional. When we do the algorithms that Erin talked about in the last presentation on variation, the SIFT and polyphen algorithms take into account alignments between species when considering the impact of variation. And highly conserved regions can be both coding and non-coding. We do find it predominantly in coding regions, but sometimes we also find non-coding regions that are highly conserved. We can also use it to troubleshoot gene predictions. We can align regions between species and say, well, that gene is there and that gene is there, so that should probably be matched up. And we can define synthetic regions. And we do both pairwise and multiple whole genome alignments. So pairwise alignments, we use BLASTZ and LASTZ-net, and we compare fairly closely related species. So we compare human to everything, and then for sort of less commonly studied species, we compare them to human and then the main model organism that they would be associated with. We also do Inredo-Peak and Orpheus, EPO analysis, for sets of species. So we have a set of fish, a set of sauropsids, a set of eutherian mammals, a set of primates, and we also do Mercator-Pekin analysis, which is done for a larger group of mammals and birds. So this is slightly a looser analysis in the Inredo-Peak and Orpheus, so you can do it for less closely related species. We also calculate shared synteny between pairs of species. So if we do an alignment, we will also look at the synteny between them. So synteny is essentially blocks of aligned regions across a larger space. So there may be gaps between these blocks of aligned regions, but it is across a larger space. In our definition of synteny, we allow for regions that have been flipped. So if something is in approximately the same region but is flipped over, we will still count that region as being synthetic. So we're going to look at a human genomic region on chromosome 2, which contains the HOXD cluster, and we're going to find alignments and conservation regions. So I'm starting again on the home page of Ensembl. I'm going to type in human and my region, and I'm going to hit go. And this takes me to this region on chromosome 2, which, as I said, contains the HOXD cluster. HOXD is highly conserved, and it's involved in limb patterning. So I'm going to go into configure this page, and I'm going to show you some of the comparative genomics tracks that we have. So I'm going to go to comparative genomics here, and I'm just going to start. So one of the things that's turned on by default is the constrained elements for the 70 eutherian mammals. I'm going to turn on the conservation score and the alignment itself for this. You'll see that there are other multiple alignments that we can turn on as well. So we've got the amniotic vertebrates, a smaller group of mammals and a group of primates. So what we have here, so we have the 70-way GERP elements. These are called constrained elements. They're called constrained because they're considered to be constrained by evolution, prevented by natural selection from changing between species. And because we consider these to be likely to be functionally important. Underneath that we have the scores, so the peaks of these were how these constrained elements were calculated. And underneath that it's the alignment, so we've got the darker pink indicates aligned, whereas the paler pink is lower alignment. If we had white, that would be a gap in the alignment. I'm going to turn on some individual alignments as well, so I'm going to go back into my configure menu and I'm going to go into blast Z alignment, and I'm going to pick a few species. So I'm going to go with something quite nearby. I'm going to go with chimp. I'm going to put that in normal. I'm going to go chicken because that's quite far away, and I'm going to go with a midpoint, which is mouse. And these are going to appear at the bottom here. So here you can see we've got alignment across the region. Again, we're using shades of pink. So we have the filled in pink indicates that there's alignment. The gaps, these white spaces, are where there is no alignment. You can see that the amount of alignment follows what we know of evolution. So the chimpanzee is pretty much aligned across the whole region, whereas the mouse, we've got a few gaps. And in the chicken, we've got a lot more gaps, which is exactly what we would expect. There are other ways that we can view alignments. We can view the alignments as text, which is listed as alignments text in this menu. And I need to select an alignment to view, so I can click on this button here and I can choose either multiple alignments. So if I open this up, you see I get my different groups, or pairwise alignments. So I'm going to go rodents, rats and mice, and I'm going to pick mouse. If I hit apply, it offers me different blocks of alignment, and I'm going to go with this largest block here. It always ranks them in terms of size, so it's usually best to go with the largest one. And if I go display full alignment, you can see I've got text alignment of the regions, and if I scroll down, you'll see that I've got red highlighting, and this shows me where the exons are. Usually you'll find that these red regions have better alignments than the regions around them. You have much fewer gaps here. If I go back up, another way you can look at alignment is using this region comparison, which is just underneath alignments text in the menu. Again, I need to select my species. I haven't got anything at the moment, so I'm going to select species or region. And you don't have the multiple alignments here, you can only pick one at a time, so I'm going to go with mouse. And what this shows, it just starts by giving me a summary of the two regions, and then as I scroll down, I have the two regions kind of lined up against each other. And we've got this alignment track shown here as well, and the useful thing is it shows us how they're actually aligned. So everywhere that this green highlighting maps between the pink, that's telling us that they're aligned. So if we get to this region over here, as we move kind of away from Hoxd itself, you'll see that there's a bit here in human that isn't in mouse. This bit in human is kind of shifted over this side in mouse, and then there's a bit in mouse that isn't in human. One thing that's quite nice about this is it allows you to configure the page for both of the species. So if I go into configure this page, I can configure either human or mouse, which means if I want to compare something that's not shown by default, if I want to, well, in this case the regulatory features are shown by default, but that's something that I might want to look at and say, well, where's the promoter in mouse? Where's the promoter in human? Is it similar? I won't change anything, I'll just save and close that. And the last thing I'm going to show you is Synteny, which is over here in the menu on the left. So this Synteny view, it's currently selected mouse for me. It shows me a chromosome down the middle, and this red highlight is the region I'm currently looking at. And you can see there's this purple highlight here, and if I follow the line from the purple highlight, it's then highlighted on mouse chromosome 2. So I know that this region, which contains my region of interest, is synthetic to a region on mouse chromosome 2. This yellow region here, if I follow the lines, is synthetic to a region on mouse chromosome 1. So you can see how the different chromosomes were rearranged over evolutionary time. If I look down, there's a table at the bottom that shows me a list of all of the genes that are found within my region. And it shows the homologues that are available between the two species. I can also go upstream and downstream to see further. So that's everything I wanted to say about comparative genomics in Ensembl. If you are interested in comparative genomics, we do have a paper that's worth having a look at. If you're working through the course, you'll be able to move on now to the exercises, which are available on the online course, and we'll post the video up on there as soon as we can. If you are working through the online course and you would like help with the exercises, you can join our Slack channel. If you're registered, you should have been sent a link to this. Or you can email us helpdesk@ensembl.org. So today's webinars were on variation data and comparative genomics. The next webinar will be on finding features that regulate genes and data export with Biomart. There are loads and loads of help and documentation available on using Ensembl. Please do check out our online course, our YouTube channel, and if you're stuck with anything, please email us. We are helpdesk@ensembl.org. And you can follow us on social media. We have Facebook, Twitter, and a blog. You can find out a bit more about what it is we're doing. And if you do use our data, please cite our publications. This is our team, our most recent retreat. And this is everybody's names and also all our funders, who we are very grateful to for allowing us to make Ensembl exist.